to the session Potpourri 2. It's one of my favorite sessions. Potpourri is a great evolution. And we're starting off with Nicole Crianza. And think about questions to ask at the end. You raise your hand or you can put it in the chat.
Well, thank you so much for having me, for being here today. I'm going to talk about how birdsong is affected by selection pressures and the relationships between cooperative breeding and female song, in particular, but male song as well. So to think about
we don't see your
birdsong.
slides yet. Sorry.
Oh, really?
Do you see them, Karen?
No. I see them. Can you see them, Leland?
I also see them. Yep.
Okay, so maybe just me. Okay.
Okay. Tell me if there's something you think I should do, Brian. I'm happy to.
No, let's go for it.
Okay, so we're thinking about how birdsong is affected by selection pressures, and birdsong is a learned behavior, and so it's particularly interesting to think about, I think, in the context of sexual selection and natural selection. How do these processes affect learning? And so thinking about the functions of learned song, canonically, birds have been thought to sing to attract a mate and defend a territory, and to identify themselves, to have to broadcast what species they are. And so this led to a set of predictions, some of which were made by Darwin, that birds with the best songs can attract the best mates, and that sexual selection would then favor showy and complex songs. The more elaborate your song was, the more likely you were to attract a good mate. And so by extension, if you're trying to attract multiple mates, that might amplify the selection. So the individual birds, if there's the fitness differentials between attracting a good mate and an excellent mate, for example, then maybe the sexual selection pressures are meaningful. But if it's between attracting 5 mates and attracting 0 mates, that the sexual selection would really have a huge effect on, and song would be really important. And so Darwin, in his book The Descent of Man, hypothesized that polygynous birds should have very complex songs, because this sexual selection pressure in trying to attract multiple mates would be really salient, and would lead their songs, that the best males would then show these ultra complex songs, and those would pass on to the best mates over time. And so we tried to quantify this in a phylogenetic framework. And so here's one of the ways that we measure song complexity is with song repertoire size, the number of unique songs in a bird's repertoire. And another one is the syllable repertoire size, the number of unique syllables in a bird's repertoire. So my graduate student, Kate Snyder, compiled the largest database on mating systems and song features that had been put together. We collated lots of different data sources and came up with 764 species where we had social monogamy or social polygyny defined. And then for subsets of those, we had different song features. And so what we found was, I think, phylogenetically very interesting, that when we accounted for phylogeny, songs did not have any more syllables or song repertoires. Those weren't larger in the polygynous species. But using one of Brian's algorithms, Brownie, we found that their repertoire size evolved faster in polygynous lineages than in monogamous lineages. And so when we quantified the rates of evolution and matched back along phylogenetic history, what we found was that there's faster evolution, but not necessarily in a particular direction, such as toward complexity, when individuals were trying to attract multiple mates. And so when we think about this from a female song perspective, and I'll talk about how we collected this data in the second half, it seems that female song appeared to be more stable with monogamy than with polygyny. So if we look at these arrows of transition, how often birds are transitioning between these states, we see that monogamy combined with female song is relatively stable, but the evolutionary transitions occur mostly toward this state, and that polygyny, when female song is present, was not very stable. That birds tended to lose that combination and go back to monogamy, or lose female song and go into this combination. So the combination of polygyny and female song absence, so polygynous species where female songs do not sing, was also more stable. That it was slower to leave that state and faster to get into it. So female song overall appears to be more stable in monogamous systems than in polygynous systems. And so most of this is published in the Nature Communications article from 2019. So what about when sexual selection pressures are not skewed toward males? What about when they're a little more balanced? So the best example we could come up with for this was cooperative breeding. Cooperative breeding is when individuals in a population cooperate to raise offspring. And so we have multiple adults raising the breeding pair, only two of which are their parents. And this happens, this has evolved numerous times. About 13% of songbird species have this phenotype. And it could be evolutionarily beneficial because in kin selection, if helpers are improving the survival of the young, the young and thus the later helpers could both be better. But it involves sacrifice of your own offspring at the time to invest in your own offspring to try to reduce that aren't reared. So the prediction we have here is that in cooperatively breeding species, a proportion of males might then opt out of the mating pool and focus on helping. And so that would decrease the sexual selection on breeding males, or by correspondingly increase the sexual selection on breeding females. And females were potentially had higher sexual selection and males lower. Potentially this balances out. This is not studied in that many species exactly how these pressures balance each other. But by extension, we might think about the song in terms of attracting mates. The song might evolve more slowly, and females might be more likely to sing in species that are breeding cooperatively than in species that are competing for mates. And so, in combination with the data that we had collected before that Kate put together in my lab, we worked with a high school student, Elena Loughlin-Pierce, and she helped us further add female song data into this database and kind of increase our sample size of cooperative breeders. And so we wound up with over 1,000 species where we have data on cooperative breeding and female song, and for different subjects of those, we also have information on male song. And so when we ask the question, do songs evolve differently in cooperatively breeding birds? What we find is similar to kind of our results with polygyny. We didn't find raw differences in the repertoire size of cooperatively breeding or non-cooperatively breeding birds. There were some trends, but when we accounted for phylogeny, there were not significant trends. However, when we go back to Brian's algorithm to think about the evolutionary rates, we found that male repertoire size seems to evolve more slowly in cooperative lineages and faster in non-cooperative lineages. And so I think if we compare the phenotypes that we saw here, that the polygyny, where we expect amplified sexual selection, led to faster song evolution, and cooperative breeding, where we expect kind of reduced sexual selection, seems to have led to slower song evolution. And so for male songs, I think these patterns kind of complement each other really nicely. And something that we followed up on and wanted to point out was that the slower male song evolution seemed to occur for both cooperative breeding and familial living lineages. So we see the faster rate of evolution is only in the non-familial and non-cooperative lineage, and then all of the lineages that had either cooperative breeding or familial living had slower evolution. And so for the male song repertoire size, what we found was that only these non-familial, non-cooperative ones have the faster rate of evolution, and everybody else is evolving a little more slowly. I think this makes sense because it's in these non-familial, non-cooperative systems that we would expect things like, territory defense, mate attraction, polygyny to be most salient, and that in the familial and cooperative context where song is maybe functioning as a slightly more social role, we might see this slower rate of evolution. And so when we ask whether male songs evolve differently in cooperatively breeding birds, we find that song elaboration, having a more elaborate song or solo repertoire, appears to evolve more slowly in both cooperative breeding and familial living situations. And so then we turn to female song again, and we think about whether females are more or less likely to sing in cooperatively breeding birds. And so this is just the raw data here. The cooperative breeding lineages are more likely to have female song present, and I think disproportionately more likely than non-cooperative breeders. So we have here, 4 times more species with female song present, where here it's a little less than twice as many species. So female song is it's common, it's widespread, it's especially for how little it's been studied and how little time it's been widely recognized. It's a very pervasive phenotype. But we have 1,000 species here represented, and we can see that the cooperative breeders seem to be overrepresented for presence of female song. But what we wanted to account for, of course, is phylogenetic history, how often these things are evolving. And so we adapted a method from Holstenbeck et al. In 2003 to try to think about the overlap along the species history. And so we make simulated stochastic character maps of these traits, and then we overlay the same tree with the two different traits, and we ask how often do these phenotypes co-occur in evolutionary history across hundreds of simulations. And then we randomize the order of the tips for both of them to say for the same proportion of female song and of cooperative breeding, how much would we expect the trees to overlap? And so what we find is that there's more evolutionary co-occurrence, more overlap in the trees for cooperative breeding with female song present than you would expect based on how often those traits occur by themselves. And so we have this red asterisk here indicating that cooperative breeding and female song presence are there. Oh, I cropped out the much more, the real data is in purple, the expected data is in green. It's more than you'd expect. But okay, I left the legend on this one, that's good. So the simulation data, when we have the randomization of the tip order versus the real tip order, and then we simulate over many, many trees, what we find is that familial lineages with cooperative breeding and female song occur together much more often than you'd expect by chance. But cooperative breeding and non-familial living and non-cooperative living, everything else occurs less often than you'd expect by chance. So the association between female song and cooperative breeding is specific to when the cooperative breeding is among kin, in a familial living circumstance. When we have cooperative breeding among non-familial, non-kin systems, female song is there less often than you'd expect. And so this is different, a little bit different from what we saw with males. And so when we look at the evolutionary transitions and how fast things are going in and out of these different states, we find that the pattern of cooperative breeding co-occurring with female song seems to be related to a pattern where cooperative breeding and female song is less likely to be lost. The female song is less likely to be lost in cooperative breeding than it is in non-cooperative breeding. And so cooperative breeding seems to be protective of female song, avoid its evolutionary loss, as opposed to making it more likely to evolve. So when we ask whether females are more or less likely to sing in cooperatively breeding birds, it seems like cooperative breeding fosters female song, particularly cooperative breeding among kin. And then when we think about these sex-specific links between cooperative breeding and song, our observations suggest that cooperative breeding stabilizes male song repertoires and also stabilizes female song preference. So I'd like to thank my lab, particularly the folks that put together this data and our funding sources. And thank you so much for listening. I'm happy to take any questions.
That's great. Henry, question. You have to unmute. Good.
Okay, am I unmuted?
Yep.
Yeah.
Perfect. Yeah. So, thank you, Nicole, for your talk. One question I was curious if you had looked into it all was, obviously you couldn't have this for the full data set, but did you look at all with a subset of the data at extra pair mating? Because obviously that's a whole nother part of sexual selection, especially with Aussie songbirds that is, and it's in cooperative breeding situations such as fairy wren. So I was just curious if you had looked into that.
Yeah, we have the Nature Communications paper that I put up in halfway through the talk. If you look at my name and my grad student Kate Snyder, that we did the full analysis on both polygyny and extra pair paternity. We saw much stronger patterns with polygyny, and so that's what I wanted to kind of do that contrast between polygyny and cooperative breeding. So I didn't focus on EPP, but we did look at it very extensively. And so in that data, we have all of our data and code are public, and the results are in that NatComs paper. But yeah, we saw stronger trends with polygyny than with extra pair paternity.
Great. Thanks. We should have Kiana set up.
Stop sharing. Thanks so much.
Okay, can you see my screen?
Yep.
Okay. So I'm Kiana. I'm a master's student at the University of New Mexico, and today I'm going to be telling you guys about my project for my master's degree. So we start the story at, oops, click, okay. We're starting the story at the crest of the Sandia Mountains in Albuquerque, New Mexico. And even though it looks nice and sunny in this photo, Audubon's warblers actually start breeding here in the spring when it's still snow-capped and overnight temperatures drop well below freezing. But yet they're ubiquitous, with population estimates in New Mexico alone of nearly 500,000 individuals. So how do they do it? The answer lies in their mitochondria. So to generate heat for survival and reproduction in these harsh environments, Audubon's warblers produce ATP through complexes in the electron transport chain. And these complexes are co-encoded by both the nuclear and mitochondrial genomes, which are under different modes of selection, inheritance, recombination, and mutation. And so these two genomes must therefore co-evolve to conserve efficiency. And this mitonuclear coevolution can result in different mitonuclear combinations within taxa. And we see this throughout the tree of life. It's evident in toad-headed agamas as elevational replacements occur throughout the Tibetan Plateau, and in killifish, where different mitochondria are present in northern, more colder waters versus southern, warmer waters. But for today's talk, I'm going to be telling you about this phenomenon in Audubon's warblers. But to understand it, we need to review the relationship with the myrtle warbler. And so both are a part of this whole yellow-rumped warbler complex, which actually includes two other birds. And all four birds differ in plumage coloration, range, and lifestyle. So here are all four birds of that complex, but today we're only going to be focusing on the myrtle and the Audubon. So myrtle warblers live at higher latitudes, migrate longer distances, and have a white throat. And their range is here in the blue. And Audubon's warblers live at lower latitudes, have shorter migrations, and have that distinct yellow throat. And their range is here in the red. With different oxidative demands at colder temperatures at higher latitudes and with longer migrations, it's no surprise that their mitochondria are also pretty different. They're actually 4.1% divergent. And if you notice, their ranges also come into contact right in Western Canada. And these birds have readily hybridized throughout that zone since their initial divergence, which was 16,000 years ago. Over time, repeated back crossings have resulted in the introgression of myrtle warbler mitochondrial DNA, so this white mitochondria, into northern Audubon's warblers populations. But not southern ones. And this introgression has occurred cryptically, as there's no phenotypic divergence between Audubon's warblers with either mitotype. So today in the Museum of Southwestern Biology, we can find these two Audubon's warbler specimens that were collected on the exact same day, the exact same mountain range, at more or less the same location. Phenotypically, they're both Audubon's warblers with that very distinct yellow throat. But one of them has the Audubon's mitochondrial DNA, and the other has myrtle warbler mitochondrial DNA. So this mitonuclear discordance in yellow-rumped warblers follows a latitudinal climb. In the north, we have the myrtle warbler with that distinct white throat, and moving southward, we see Audubon's warblers with the myrtle mitochondrial DNA. And even further south, we see the Audubon's warblers with the Audubon's mitochondrial DNA. So right here in the Southwest, these two mitotypes co-occur cryptically in what are phenotypic Audubon's warblers. So what caused this introgression to stall? Is there some sort of selective advantage to having the myrtle warbler mitochondrial DNA in the north, but not in the south? These are questions that have been asked before, specifically by Dave Taves in 2014. He sampled an eastern and western transect throughout this mitochondrial contact zone and found the centers of each transect. He also collected feather isotopic data, which suggested that discordant Audubon's warblers are migratory and non-discordant Audubon's warblers are not. This then was proposed as the mitochondrial the migration adapted mitochondria hypothesis. And under this hypothesis, there's a migratory divide where discordant Audubon's warblers in the north would migrate over non-discordant Audubon's warblers in the south. And this would result in a leapfrog pattern of migration, where resident birds in the southern end of the contact zone would remain year-round, while migrants would move back and forth between seasons. And under this migration adaptive mitochondria hypothesis, wintering birds in the resident range of the contact zone should resemble breeding birds because they're not migrating between seasons. We decided to test this hypothesis by comparing wintering and breeding birds' plumage and mitochondrial composition using wintering specimens from New Mexico. So in the breeding season in New Mexico, we see what are only phenotypic Audubon's warblers with that bright yellow throat. There's absolutely no phenotypic structure. So to assess wintering birds, we scored the plumage characters of 42 wintering adult birds following Hubbard 1967. We used a hybrid index to score the throat color, the auricular patch, and postocular line, with scores of 0 indicating the myrtle phenotype of the white throat, the black auricular patch, and the full and distinct postocular line. Scores of 2 indicating the Audubon's phenotype, which is the yellow throat, no auricular patch, and no postocular line, and scores of 1 indicating any intermediacy. We also used a numerical score to account for the amount of white in the tail, ranging from 0 to 1, which is shown here. And we ran a PCA and cluster analysis on these plumage scores. We found 4 distinct clusters, with PC1 accounting for about 51% of the variation, and was primarily loaded by throat color and the amount of white in the tail. And PC2 separated about 27% of the variation and was loaded primarily by the auricular patch scores. So this composition of 4 unique plumage groups is in complete contrast to the lack of phenotypic structure that we see during the breeding season. We then decided to look at the mitochondrial DNA for wintering birds, and we found that all 45 birds sampled had myrtle warbler mitochondrial DNA. So the star here indicates the center of the cline that was found in Taves et al. 2014 that I showed earlier, which is where both mitotypes occur at 50% frequency. So to find localities below this center in New Mexico with solely myrtle warbler mitochondrial DNA suggests that the birds wintering in New Mexico are actually different from the birds that are breeding in New Mexico. So when we revisit this migration adaptive mitochondria hypothesis, we don't find support for wintering birds resembling breeding birds in either plumage or mitochondrial DNA composition. It appears that breeding birds are in fact migratory and being completely replaced by wintering birds from further north. So if it's not migration shaping this mitochondrial contact zone, what is it? We bring about this new temperature adaptive mitochondria hypothesis, where the mitochondrial DNA cline is associated with climatic variables. But to test this, we first had to visualize the contact zone, and using 593 birds, including 368 museum specimens and a Kriging interpolation approach, we did just that. We found that discordance increases in frequency moving northeast throughout the contact zone. So we can see the frequency of each mitotype at localities that are represented by the pie charts throughout the contact zone, with white indicating the myrtle warbler mitochondrial DNA and yellow indicating the Audubon's mitochondrial DNA, or discordant and non-discordant birds. We plotted the center, which is where both mitotypes occur at 50%, and that's shown by the sigmoidal curve running through the middle of the plot, and the orientation of the cline, which is shown by the double-headed arrow. The cline deviates from latitudinal bands, suggesting that it's shaped by more than just latitude alone, which was what was previously thought. We then repeated this analysis using individuals instead of locality clusters, and we found nearly identical results when it came to cline center and cline width. We also compared our center to those found in Taves et al. In 2014 and found consistency, with a higher center in the west than in the east. But now we have a much more detailed view of what's going on between these initial 2 points. This also shows that the contact zone appears to be stable and doesn't seem to be moving even after a decade. We then ran logistic regression models to determine if any fine-scale climatic patterns improved the predictive accuracy of the Kriging, but we found no significant differences between models that included the Kriging and any additional climatic variables. We did, however, find strong correlations with the Kriging interpolation and temperature, which here is PC1 of bioclimatic variables, precipitation, which is PC2 of these bioclimatic variables, elevation, and latitude, suggesting that the mitochondrial distribution, these Kriging interpolated values are associated with these climatic variables. So going back to the temperature adapted mitochondrial hypothesis, we found some weak evidence of climatic association with the mitochondrial distribution, but we also may just be looking at too broad of a scale to determine any finer patterns. So in conclusion, we have shown that the composition of wintering phenotypes is different from that of the breeding birds. In the summer, we only see the Audubon's warblers, whereas in the winter, we see Audubons, myrtles, and hybrids, which can only come from that hybrid zone found in Western Canada. We've also characterized the distribution of breeding mitochondrial DNA throughout the context zone and found its center and orientation. We also found some weak supportive evidence of climate, not migration adapted mitochondria. So what's next for this system? We know that breeding birds move around, and within days of fledging, an individual can easily scale different elevations or different slopes. So we don't really know the exact location of where these birds were breeding, only where they were collected. So we want to eliminate some of that noise by looking at precisely where these birds are breeding, also known as their nests. Are discordant birds with the myrtle warbler mitochondrial DNA breeding earlier than non-discordant birds? Because in theory, this myrtle warbler mitochondrial DNA tolerates colder temperatures and higher oxidative stress. Are discordant birds able to tolerate these earlier, harsher conditions? We think so. So we zoomed in on one slope of one mountain range and searched for nests all throughout last breeding season. And we found, sampled, and aged 10 nests from the eastern slope of the Sandia Mountains. And we found that discordant birds, those with the myrtle warbler mitochondrial DNA, seem to be breeding about 5 days earlier than non-discordant birds. Is there a physiological advantage to having myrtle warbler mitochondrial DNA? Are discordant birds just arriving earlier? We don't know the exact mechanism, but as of actually this morning, we have added 20 new nestlings to this sample size to test if these results are robust. So stay tuned because our team is actually really excited about these preliminary results. And so with that, I have so many people, agencies, funders, and museums to thank. This would not have been possible without any of these people or agencies listed here. And also I want to acknowledge the museum specimens that we used. Some of these birds were collected before I was even born, so this project would not be what it is without these 368 museum specimens. And with that, I will take any questions.
That was great. People feel free to raise your hand or just enter questions in the chat. One question I have, so with opsins, people can go from DNA sequence to predicting function. Can the same thing be done with mitochondrial DNA, where you could say, let's sequence the genome of this mitochondrion and say, this is really good at high temperature, things like that?
We could do that. We actually didn't sequence the mitochondrial DNA. We just used an RFLP assay because there's a SNP in one mitochondria that we have a restriction enzyme to be used for this. So it's a lot cheaper to do that, and that's what we did. But we could in the future sequence the whole mitochondrial genome and see if there's different functional assays between them.
Right. Other questions? Oh, we should let the next talk set up. Amy, go ahead.
Hello, so my name is Amy Ventura, and the project I'm presenting is image classification. We created an AI model to identify hybrid, juvenile, and adult trout. So introducing species into an area creates novel opportunities for hybridization with the native species, and in some cases, this results in the native species declining from hybridization, which is sort of the case in our species, where we have the native Yellowstone cutthroat trout, and they are declining in part due to hybridization with introduced rainbow trout. Rainbow trout have been introduced broadly throughout North America, and so where they overlap with the cutthroat trout subspecies, they often hybridize extensively. And so what I mean by hybridizing extensively is the F1s are fertile and the parental species persist in hybrid zones. So we have widespread introgression. This means basically any kind of hybrid you can imagine, you're probably going to find them in this system. These are all the individuals used in this project. So each dot is an individual, and where it's located on the graph gives an idea of its ancestry. So the x-axis is a proportion of Yellowstone cutthroat trout ancestry. If it had 0, then it's rainbow trout, and if it's at 1, then it's Yellowstone cutthroat trout. The y-axis is interspecific ancestry, which is a portion of the genome where each loci has one gene copy from one species, another gene copy from another species. And so if you have low interspecific ancestry and then a high proportion of Yellowstone cutthroat trout ancestry, then you are Yellowstone cutthroat trout, and the opposite, you're a rainbow trout. And then if you have about 50% rainbow trout, Yellowstone cutthroat trout ancestry and high interspecific ancestry, you'll be an F1. When the F1s interbreed, recombination results in a lower interspecific ancestry. So that's where the F2s will be. And then when all the hybrids interbreed with the parents, you'll get the different back crosses. We also have a other category, which is just like a catch-all for individuals that don't perfectly fit within the other categories. And so one of the goals when it comes to conservation is correctly and consistently identifying the species of interest. And so you can imagine that we have complex hybridization dynamics like this that also complicates morphology. So these are the two parental species. There's Yellowstone cutthroat trout on the top and rainbow trout on the bottom. And so these they're pretty distinct from each other, and they have features that we use to differentiate them. For example, Yellowstone cutthroat trout, they have that namesake red slash. Rainbow trout don't have that. Yellowstone cutthroat trout also have fewer dots on their head. Rainbow trout tend to be more spotty. And rainbow trout also have a white leading edge on their pelvic and anal fins that you don't see in Yellowstone cutthroat trout. And so for humans, we're pretty good at identifying these. We have about 100% accuracy of differentiating them, but things do get more complicated when you get hybrids. So these are F1s, and what you would usually expect is F1s will be consistently intermediate from the parentals. Unfortunately, that's not what we see with these F1s. So you can see from the top that the top F1 has a red slash and a very slight white leading edge on that pelvic fin. And the bottom has two white leading edges, very few spots on the head, and no red slash. So they look fairly different from each other. And then these are more F1s, and you can really see the variation here where you've got the one F1 on the bottom has no white leading edges, and the one on top only has one. Back crosses are slightly more consistent. You would expect them to look more like the parentals, but we still get a degree of variation within them. So for example, the Yellowstone back cross on the bottom does not have that cutthroat, even though it looks more like a Yellowstone cutthroat trout. And the rainbow trout back cross on the top only has one white leading edge. And you see it with this other rainbow trout back cross where it only has one white leading edge, where an F1 might have two. And then this back cross on the bottom has the red slash, but it has the coloration more of a rainbow trout. And so my point is basically it's very difficult to identify these fish. The outcomes of hybridization are like broad. And so for humans, we have about a 75% chance going off of specific features to identify a hybrid from parental. And so it actually, we haven't tried it on juveniles because juveniles are so much more difficult to ID. Anybody who's taken a professional fish identification course, and yes, they exist, knows that juvenile trout are sort of a class of their own where they're smaller, they have less features than adults, and they have different features from adults. So we have not attempted in this system to identify juvenile rainbow trout, juvenile Yellowstone cutthroat trout, or their hybrids. We have noticed though, that a white leading edge on the dorsal fin is a pretty good indicator of some degree of rainbow trout ancestry. And so that leads us to hope that there are some features that will differentiate these two. And so that leads into my question, where we have on one hand, human identification where it's fairly unreliable, it takes a long time to train people. On the other hand, we have genomic identification, which is very reliable, but as I'm sure most of you know, it's quite expensive, requires technical equipment and isn't very accessible to regular person. So we were wondering, with AI becoming so much more advanced, can we use computer vision to find a middle ground between these two systems? And so we have all these photos. Each photo that I used in this project is attached to a genotype trout. And so we have about just under 2,000 trout and just under 3,000 photos. And so by genotype, what I mean is my colleagues did genotyping by sequencing and then used a structure like a genetic clustering model to estimate ancestry, which is along the x-axis here. And you might have noticed that for both the adult on the left and juvenile on the right, our sample sizes are skewed towards rainbow trout back crosses, and a little bit to the F1s. And so this is just sort of the unfortunate reality. Rainbow or Yellowstone cutthroat trout are declining, and they're getting harder and harder to find. That's just a limitation we can't get over. But one limitation that we can get over is small sample size. And so that might be surprising to hear, because usually these sample sizes are really good, and they are really good for population genomics and hybridization research. But AI usually requires a little bit more photos. So most animal classification models are trained on hundreds of thousands to millions of photos. And in comparison, 753 and 1700 is kind of like pennies. So what we did was image augmentation to boost our sample sizes. Basically, what I did is I took the original photo, I appended the machine, I just told it to do something simple, and it output all these pictures. Mostly I just asked it to rotate, do a little bit of zooming in, flipping the photos, which to you and I, just looking at it, like these are obviously the same photo. There's not much of a difference, but because the pixels move around, the colors change, it gives the machine a better idea of what pixels are important and therefore what features are important. I'm going to get into model training now. I'm going to just skim over it. I have gotten very familiar with this model, so if you have technical questions, I'm happy to answer them later. But basically what I did was I separated all of our photos into 70% for training the model and then 30% for testing the model. And so these training photos are the ones that got the image augmentation. I pass it off to the model. I say, look at these, here's the ancestry, learn what these photos and this ancestry means. And it does that about 55 times. It goes through the photos. And then at the end, I pass it these 30% photos. It has never seen these photos before. These are brand new. And so it has to use what it learned looking at the other photos to predict the ancestry of these brand new photos. And so it gives us that. It says, I think this is what they are, and we can compare it to what they actually are. And so important one thing to note before I get into this is that each of these heat maps is its own model. They are trained on the exact same photos, but that does sort of explain some of the number discrepancies here. But the one hybrid model I find it's more accurate, but the 5 hybrid model I find explains some of the errors in the one hybrid model that we wouldn't see otherwise. And so all of the individuals that are in the yellow boxes are correctly identified, and everything outside was a misidentification. And so you can see that there were no mistakes between the two parental species, which is excellent. This is what you'd expect for adults because they're easy enough for humans to identify. You should expect an AI to do it. And then you also see that there is no errors between the two kinds of back crosses, which is also excellent to see. That makes biological sense. One thing I find interesting is that if you look at the hybrid parental misidentifications, which are mostly between rainbow trout and their hybrids, which when humans were identifying them, they also found that problem too, are mostly between, back crosses. So most of the errors were just between rainbow trout and their back crosses. And then another thing I find interesting is that when they were incorrectly identified, they were usually identified as a back cross rainbow trout. And if you remember that triangle plot from earlier, the other category is sort of catch-all, but for the most part, these fish skew towards rainbow trout ancestry and are kind of similar genetically to back crosses. So I find that interesting. And now's the cream of the crop. I'm so excited that the juvenile model worked. We almost weren't expecting it to work, but we got almost the exact same results with the juveniles as we did with the adults. And so you can see, again, 2 different models. If you're in a yellow box, you were correct. We see no errors between the 2 parental species, which is fantastic. There are some errors between the 2 back crosses, and there's more, a couple other things are different between the 2 models, but it's really good to see this. And the other thing that's very different is between the hybrid parental misidentifications with the rainbow trout, there's more variation. So it's still mostly back crosses, but you see more like the other F1s, a couple F2s there. And so we are suspicious that It might be due to the juveniles being very small and hard to identify. It could also be due to a larger sample size, because just as you raise your sample size, you will get just naturally more variation, you'll get a unique phenotype that's harder to ID. And another possibility is that juveniles are very small. Some of the photos were kind of low quality, and so I haven't had the chance to like go through all the photos yet. So it might be the errors are just like the fish looks like a stick. We can't tell what it is. But overall, I would say that our model was super successful. It was capable of identifying juveniles. It made no mistakes between parentals. It was really good at isolating Yellowstone cutthroat trout from the others, which is pretty useful because Yellowstone cutthroat trout is the native species and the hybrid and the rainbow trout are sort of introduced. We did find, as expected, backcrossing complicates phenotypic identification. It's genetic identification is never going to be eliminated. We weren't aiming for that. But the good news is that if you can tolerate some degree of error, if you have a project where you don't need 100% like this as a backcross, you can just deal with a little bit of error. You could use this program in this system or other systems to track degree of hybridization over time, to isolate the native species from the hybrids and the introduced species, which could be useful in systems where they're actively being removed. And another thing is, specifically in this system, when you catch juveniles and you take tissue samples from them, it's usually lethal. And so in this case, if you want to track young of the year, a degree of admixture, you could use this program in this system and potentially other cutthroat trout systems. You'd have to train to the model, but you can use it to get an idea of what's going on with juveniles. I'd like to thank everybody who's part of this project, who caught the fish, who did the sequencing, the clustering. I really appreciate it. I have done it. It's a lot of work. I will be at Evolution Montreal sharing my master's project, so I hope to see you all up there. Is there time for questions? I can't see the time.
Yep, we have time for questions. So, folks have questions? I can start. So oftentimes with machine learning stuff, people take like a pre-made model, like an iNaturalist vision model, and then train on that. Sound like you did de novo training. Why did you make that decision?
So we started off with a pre-trained model. We were using Resin 18, so we just used like their generic train model. But for this one specifically, we really needed that genetic information. So we could have shown it, like, I know that iNaturalist has a whole collection of trout photos. We could use those, but we wouldn't have that backup of knowing what their true ancestry is.
Oh,
so
yeah,
I think that's
the
useful part of this.
Cool. Other questions? Wasn't it time just to follow up? So one of the people will do is they don't use that model as the validation model, but they use it to help inform their new model. So like they still do the 70-30 on a model, but they start with that model and they add to it using the training.
Oh, yeah,
so,
we did do that.
Okay. Yeah.
Sorry,
Cool.
I misunderstood.
I wasn't clear.
Yeah, I have a question about the back crosses. And so like just if you were to do a genetic analysis just because of recombination and stochasticity, those back crosses are going to be much more genetically variable than say an F1, which is very repeatedly a heterozygote of the two things. So do you think that maybe some of the more variability, the more errors that you're getting in that back cross thing is just really picking up genetic based phenotypic differentiation that's just more variable between back cross individuals overall?
I definitely think it's possible. I haven't explored, so there's different so there's sort of different levels of interspecific ancestry, and so we can compare the rate of misidentification, the direction of misidentification to that. I haven't done that yet, but I'd be interested to see if there's a correlation there where lower interspecific ancestry changes the direction and type of misidentification.
I mean, I was just kind of thinking, I actually think it kind of says that your model's even better than you think it is, because it's picking up on the variability in back crosses that that's biologically real. So it's like it's actually really picking up on the phenotypes even better than we would expect. So I just thought it was kind of interesting.
We should move on to the next talk. Jason on digital organisms.
Is Jason here? I thought he was here, but
Him
I
right
don't
now.
see him now. So okay, I'll go do a quick check by email, see if
Okay.
he's got an access problem. So there's no notification on the email. Everyone, we're just waiting for our next speaker to show up. And Jason, if you're in the audience and just with a different address, can you just raise your hand in case we've missed you? Otherwise, we will break until the next session, but we will give it a few more minutes to start and go through.
And Spencer, since people might be like coming from for all the sessions for your talk, we shouldn't start yours early. So
Yeah, something sounds good. We'll start on time,
Yeah.
no chance of overrunning. That sounds good to me.
And I'm just creating a slide so people know what's going on. With the assumption that they are not showing up.
Yeah. Very official, Karen. Nice job.
Yeah, it helps to have all the slides already open in my background and just copy over something that hopefully wasn't important. You know. Spencer, you're up next, right?
Yes, they are.
Yes, I'm all set.
Yeah, no worries. Grammatically incorrect, though. Now I have to fix it.
So thanks for all the speakers for being on time. It's really nice to have like not to like shut people down and things like that. Good.
Yeah, so far, at least the sessions I've been in, it's been really, everybody's been really conscientious about that, which has been nice. Because otherwise I just, you feel bad cutting into people's talks.
We used to have lightning talks, and I wrote an app to like have a screen display that would show like how many minutes left and the next talk up and a big red sign. But we don't do lightning talks anymore, unfortunately.
Yeah. I mean, it's hard to fit into like a lot of information into a 1 minute or 5 minute.
If I it worked okay, because we'll just eliminate the like ever since Darwin part and just get right to the meat. But I think partly it was like, it was in even more potpourri than these sessions where it felt sort of random to people, even though we tried to organize them well. And so
I'm really liking this session.
Yeah.
I have a soft spot for both birds and fish, so yay.
but Drosophila is also great.
Of course. Well, clearly, I didn't mean I didn't mean to leave Drosophila.
I know.
That was a given, clearly. Although they are the bane of my compost pile, so
You put little sundews on top or something.
be like little presents. I'm always just amazed at how quickly fruit flies find my compost bin, like in the spring and summer. Maybe
Yeah,
that's-
they're really persist, like they're good at finding.
where do they come from is what I want to know, because really, where do they come from?
But what's their overwintering phase? Like they overwinter as well, yeah. Spencer.
There is a polymorphic gene on the third chromosome that allows them to go into reproductive diapause, and it's more common in northern populations than it is in southern. But really cool is that it's a super old allele, or not super old, but it predates their movement into temperate zones. It's
Oh.
actually what they think is it's an old allele for kind of dietary restriction survival, even when they were in sub-Saharan Africa. So it kind of segregated ancestrally. And then when they started expanding out into temperate areas, it became kind of really adaptive for temperate overwintering. So this is amazing. The females will mate in like late October.
Huh.
Store the sperm. All the males will die over winter, I think. And they actually storm the sperm in the spermatheca and then use that to fertilize the eggs like in, April.
Huh.
Incredible.
That is
neat.
pretty impressive.
And
And
so
then they go straight for Karen's compost. Sorry.
Oh, I'm sorry.
I said, and then they go straight for compost.
Yes, they
Oh,
do.
yeah.
They go straight for my compost, and I curse them. Oh, but I do have a fish question. So I used to work with lab tech in a lab that studied sticklebacks, and so have you tried it with sticklebacks yet? The little because the hybrids on those were it was tricky sometimes, and it got trickier over the years. So with the benthic and the oh my gosh, I can't even remember the ones that hang out in the open water.
Limnetic.
It's been 20, thank you. Limnetic, it's been a while. It's been a hot minute. So like 25 years. So
Stickleback are insane. Like, what a pick for a model species, eh?
I love them. They're right up there with Drosophila. Sorry.
I would love to. I think they could probably apply to many systems. This is the only one I know of so far. Just because they were catching all these trout, and so one of the PIs had an idea. What if we took pictures of all of them? And so I wonder if other people have done that too. There's like someone has a cache of all these stickleback photos.
Oh, yeah, I'm sure. I'm sure we do. Sure they do. How are we doing for time? We've got 3 minutes. Okay.
Other cool natural history stories folks want to share in our downtime?
I actually want to hear more about the fruit flies, and I don't think I've ever said that before in my life. Sorry, Spencer. Sorry.
What was that?
He wants.
I said I want to hear more about the fruit flies, and I don't think I've ever said that in my life.
Yeah, no. I mean, I think that most fruit fly studies bore me to tears too, because there's not a lot of natural history in them. But melanogaster, there are a few people that do some very cool kind of natural history type stuff. I don't know. I don't really have any good stories. I do kind of boring lab work. So, but there are people. Let's see. What's a very interesting thing? So there's another gene which probably is associated with this reproductive diapause. I'm not 100% certain, called Methuselah. And if you know your biblical allusions, this is what it does is it super downregulates metabolism, and these flies just live for months and months and months and months and months. And so it looks like there's a lot of stuff that turned out to be really adaptive for this kind of become a cosmopolitan species stuff that had a lot to do with just being able to be okay with starving for a long time. And I imagine that that's not really a Drosophila thing. Like, if we were to study all the other insects as intensely, I bet there's a lot of kind of like polyphetism there associated with like just hardcore starvation resistance, like kind of segregating even in tropical populations. And I think that would be a really cool thing to study outside of melanogaster. My guess is that all the like the subobscura and the pseudoobscura, I imagine they probably all diapause. So it's not even a polymorphic gene. Like that's just what they have to do if they live in northern latitudes.
We should probably start your actual
Oh
talk.
my goodness! Yeah, sorry. This is yikes. Let's see. I'll share. Yeah, that was silly. Okay. And 1 second. All right. Does that look good to everybody?
We see presenter view.
You see presenter view. Okay. So really quickly, let's swap displays. Now, how's that?
Perfect.
Okay, great. Okay, so I'm going to tell you about inversions, and actually my talk is going to be using Drosophila data, but it's not really about Drosophila. So I think that most people are finding out that there are inversions everywhere. And like this is really an exciting time to be studying inversions because it's not just flies. These are just a few of the organisms where inversions are associated with a fascinating range of things. So there's chromosomal inversions that help promote adaptation, sex chromosome evolution, species differences, and maintenance of supergene complexes. Real cool stuff. But we also know that inversions cause fitness under dominance, have a large genetic load, and the vast majority of them go extinct in less than 20 generations. So like they're bad. And this kind of seems like a paradox, but there's a really easy resolution. It's because inversions are neither intrinsically good, nor are they intrinsically bad. In fact, they really only do one thing, is they alter meiotic recombination in the heterozygous state. And that says like everything that's positive or negative that's associated with inversions is in one way or another a product of reduced crossing over. And if that's true, then the key to understanding the evolution of all inversions is understanding how they suppress recombination or alter it both locally and globally. And that's what my study's about. That's what it talks about. That's what I kind of think about all the time. And that really is not a fly thing at all. It just so happens flies where we have the best data. So what I'm going to do is I'm going to show you first that inversions don't just suppress recombination locally, they redistribute it globally. And I'm going to use a meta-analysis to describe the redistribution onto non-homologous chromosomes. And that's what we call the interchromosomal effect. And finally, I'm going to show you that even though these global changes compensate for the local suppression on a single generation scale, their evolution is like really not that important. Right? It's actually a very small effect when compared to the deleterious mutation accumulation near inversions. So there's kind of a couple twists that are coming, but this is the talk. So here's the karyotype of Drosophila melanogaster. It's got an acrocentric X, 2 metacentric autosomes, and there's a dot that's non-recombining and the Y that's non-recombining. So we're going to ignore them for our talk today. In absence of inversions, this is what recombination's landscape looks like, and it's fairly typical. It's got this mid-arm peak of recombination intensity and low recombination at centromeres and kind of low at telomeres. When we introduce an inversion, a heterozygous inversion, importantly, like IN2RNS, the formation of dicentric chromosomes acts to suppress recombination. And that's that inversion loop diagram you see in all your textbooks. And even though it's the kind of classic mechanism, it only actually explains 10 to 20% of recombination phenotype. And that's probably a surprise because everybody loves inversions, but we don't know what causes the 80 to 90% of the phenotype. And like when I figured this out, it kind of blew my mind. And I essentially haven't recovered yet, but this talk is going to help a little bit. So first things first, because we can use compound chromosomes in flies, that is take 2 arms and attach them to a common centromere, we can actually recover single exchanges. And as far as I know, flies are the only organism we can do this. And when we do that, we actually see that recombination inside isn't completely absent, but it's way lower than what we would expect. And it's shifted away from the inversion breakpoints. We also see that recombination is suppressed outside of inversions, and there's no reason that dicentrics could possibly cause this. And it kind of decays with distance as you move away from the heterozygous inversion breakpoint. Even more surprising is if you look at the other arm, that actually the rates are a little bit higher. And really crazy is that if you look at completely unlinked chromosomes, chromosomes that have nothing to do with the inversion, for some unknown reason, the rates are actually higher as well. And this green pattern is what we call the interchromosomal effect. So if you look at the per base effects, the strong local suppression is by far the dominating force. Red arrow is the biggest. But because the weak global increases happen to so much of the genome, to a first approximation, it actually kind of balances out. And it's almost like there's crossover homeostasis acting. And I say almost because the necessary experiments of measuring all these effects simultaneously for a single inversion just simply does not exist in the literature. And what I've been showing you is like actually a cartoon that kind of takes a whole bunch of different studies, looking at different areas, put them together in a single model to build that set of predictions. So it kind of gets us to what my question is. How do inversions alter a combination on non-homologous chromosomes? And the way that I'm going to answer that is with a meta-analysis. There's not one single experiment that I can do to answer this question definitively. So let's do a meta-analysis. And once that's done, we're going to ask, well, now that we kind of have a general model, what is the evolutionary consequence? And to answer that question, I'm going to talk about forward time population genomic simulations. And that will be, I think, a kind of interesting future direction for most people as well. Okay, so meta-analyses are a little boring, but I do want to tell you that like there are hundreds, I mean hundreds of interchromosomal effect experiments out there. They usually publish the raw data, especially the old ones. And we already have the statistical framework to reanalyze that data. And like this is pretty sophisticated statistical framework. It's point processes that can be fit with maximum likelihood. Like, so I'm pretty happy about this sort of stuff. The crazy thing is, even though we have all this data and we have all this kind of statistical machinery, you really, at least I didn't before I started the project, have a good idea of what the patterns and the effect sizes were. I just kind of knew up, down, about it. So this makes it, I think, really ripe for a meta-analysis, right? You've got lots of information, you've got statistical framework, and you've got essentially an open question that's quantitative. So I'm showing you about 100 experiments, all on the X chromosomes, and all these studies were pre-1966. And the good news here is that recombination intensity really is higher on the sex chromosomes when there's heterozygous autosomal inversions around, right? An inversion on one chromosome actually makes the sex chromosome recombination rate goes up. There's a lot of variation here, but we're going to address that in a second. There's another claim that you see a lot in the literature, which says the centromeres and the telomeres are more sensitive to the interchromosomal effect. But when you actually look at the data, there's not a lot of information that substantiates that. And especially when you start using mixed models to take into account that some of these really high estimates are all coming from the same experiment, there's really just no statistical support for the differential sensitivity along the chromosome. And also, sometimes people say that the interchromosomal effect alters the crossover interference. Again, this is kind of a problem of incorrect statistical analysis, because when you actually look at a coincidence function, there's just really no evidence that the interchromosomal effect is any different than a standard homokaryotype. So returning to the one graph where we really do have a large effect, is there's tons of heterogeneity here. And really what that turns out to be is that in each different experiment, people are using between 1 and actually 7 inversions to induce the interchromosomal effect. And there's a pretty straightforward relationship. More inversions means stronger interchromosomal effect. So we can actually account for this. We can take the slope of this line using a meta-regression, and we can actually go back to our kind of 3 claims, and we can say, okay, there is an increased overall crossing over on non-homologous chromosomes. It's kind of bizarre, but per heterozygous inversion, it's about a 0.5 centimorgans per megabase increase. It's fairly uniform along the chromosome, and it really doesn't alter crossover interference. And that's consistent with kind of this local global balance of crossing over. I don't want to make a strong claim about homeostasis, because to actually do this, you need to have very precise calculations. And right now we're kind of doing back of the envelope type stuff. But at least we're consistent at this point. So instead of going into those calculations more thoroughly, what I want to do is talk for the last couple minutes about the evolutionary consequences. So once we have this number up here, the 0.5 centimords per megabase, and we can feed it into here, what is the evolutionary significance of this, right? Increased recombination should matter. So to see this, we'll use background selection, and the effect of which can be quantified by examining the ratio of coalescence times for alleles at a neutral site when background selection is operating, divided by the coalescence time without any interference, and that is B. And the really great thing about this measure is that it's actually just determined by the rate of deleterious mutations, actually the density of deleterious mutation rate over genetic map length. And if you remember back to the very beginning of my talk, inversions don't actually do anything. So they're not really changing the deleterious mutation rate, but they certainly are changing the genetic map length by reducing recombination. So this is actually a very fortuitous thing for me, because I've got a really ready-made measure. And we can look at this right now. This is the genome-wide for all standard arrangement, the homokaryotype in flies. And it actually says if no background selection or background selection was unimportant, the dot would be up here. So genome-wide, there's a pretty big effect of background selection on a genome like Drosophila. We can take this and break it up into the different chromosome arms. And they're a little bit different because each chromosome is a different size and different genetic map length. But what we can do now is we can say, what happens when an inversion is introduced on one arm? Well, that strong local suppression essentially takes you down from that like 0.66 all the way down into like Muller's ratchet area, where essentially it's almost like an asexual reproduction down here, almost no recombination. And those global increases, which were kind of compensating on the single generation scale, all they do is just kind of nudge the background selection effect up. So it's not absent, but it's just so minuscule by comparison to the deleterious or the effect of deleterious alleles accumulating inversions. Now, I personally think this is an understatement of the inequality, because with such strong local suppression, Muller's ratchet will start operating. And I haven't actually incorporated that into the simulations yet. So what's happening is the deleterious mutations accumulate, they become a serious problem. And what we really need to do is we need to explore that with forward time population genetic simulations. And we need to do it on very realistic genomic architectures, because we want to actually compare this to data that we have. So I don't have that. I'm not going to go for another 5 minutes. But what I want to say is, look, we've got this kind of interesting effect. It's not well characterized, but that is consistent with a local global balance. Now, just by definition of altering recombination, we know that we've decreased Hill-Robertson interference. And we know that we, by definition, have decreased genome-wide mutational load. But it certainly is nowhere near compensating for the accumulation locally at inversions. And what I really hope is that this has kind of whetted your appetite, and you'll come see my talk in person at the conference next month. And I'll have, I think, some new results for you on just how bad the deleterious mutational load actually is inside the inversions. And so with that, I'd just like to thank, of course, the funding from the NIH, the co-author and lab members, Dr. Seveson and all the members of the Seveson lab. And then also I'd like to put a big plug in for Dr. Alan Wilson and the students of FISH 7350 at Auburn. This is a wonderful course for all sorts of people across the university to learn meta-analysis. And it certainly has changed the way that I've thought about a lot of projects. And with that, I'll take any questions or comments, anybody has anything to say.
Great, thank you. Technically, we don't have time for questions, but this is the last talk in the session. So people can ask questions anyway. Yeah, it was great. So I have a question. So like, I mean, Drosophila works famous for having lots and lots of data and lots of people working on it. So meta-analysis doesn't seem like it seems like an obvious thing to do. People aren't doing it. Do you have a sense of why?
Yes. Well, personally, I feel that I came from a fairly statistical background, and my impression is that most Drosophila geneticists are not statistically oriented. And I think that it is of a nature of a view that essentially you can do the perfect experiment and that solves the problem, period. And for a lot of things that's done it. And so like they weren't wrong in the past. But this is one of those questions that has kind of just persisted. So the first interchromosomal measurement was probably like in the 1916s. So like we're talking 100-year-old problem and has persisted. So at very least, I can say, you know what, maybe this is the one thing that meta-analysis should really be accepted. I am 100% a convert. I think I'll be doing more meta-analyses in the future because I think it's very useful, especially with this sort of database.
That's great. Other questions? All right. Well, thank you all the speakers in the session. This is great.
Yes. Thank you.
Thanks, Brian, for moderating. Thanks, everyone.